Welcome to Dateline Health. This is Fred Lippman coming to you from Nova Southeastern University. This is a program that I've wanted to do for quite a while. We've had communications from folks in our community. Obviously it comes about because of the COVID crisis that we're going through with this insidious viral incursion into our communities. But I've been wanting to get some of the leadership from the Scripps Research Unit that is world-renowned. It's a preeminent research venue that's been in California for years, but it's been in the state of Florida for nearly 2 decades. So let me welcome to you from Scripps Research Florida, Doug Bingham, who is the Executive Vice President, and I guess they say Florida site head. I don't know what that means, but I know you're the boss. So welcome, Doug.
Thank you very much. I'm glad to be here.
Good to have you, sir. We just before the broadcast, Mr. Bingham and I were reminiscing because I was there at the very beginning in another part of my life because of my public service to move to entice Scripps to come to the state of Florida. And we're very proud of the fact that Scripps is in Florida. Thank you very much. And also we have Dr. Tina Izard, who is a professor of structural and computational biology. Welcome, Dr. Izard.
Thank you for the opportunity.
Well, thank you. Tell me about yourself. Tell me what you're, where you're from, and I think you're from Switzerland, if I'm not mistaken.
I was born in Switzerland, that's right. And then I moved around quite a bit. I got my undergraduate training in Switzerland, in Basel, and then my Ph.D. in Australia, in Melbourne. And then my fellowship was postdoctoral fellowship was at the University of Washington, Seattle. And I moved quickly to England. I was at St. Jude Children's Research Hospital when I was recruited here. And I've been here for a very long time, actually, almost 14 years.
Well, we're very proud to have you as part of our population here in the state of Florida. You are so well known in the world of computational and structural biology. And I know that you, a very important thing about what the folks here have asked about is some of the work that, that somebody wrote to me about your involvement in breaking down the molecular structure relative to the spread of cancer. You want to talk about that?
Yes, absolutely. So we just finished, after about 2 years, we just finished setting up a state-of-the-art microscope, first in the nation, that allows us to actually see the proteins involved in these biological processes at atomic level. So we can visualize things as small as water in 3 dimensions. So this is really exciting and allows us to see how antibodies, for example, bind to the new coronavirus. And this technology that won a Nobel Prize in 2017 is a huge advance for structural biologists like myself. And we apply that to many proteins, and one of them is a protein called integrin, you might have heard of, which is involved in binding to the spike protein of these viruses. And it's also involved in protecting cardiac myocytes from cardiac protection, provides myocardial protection. So we're working on a new inhibitor that inhibits these integrins that allows us then to treat this myocardial infarction, for example. And these are also important for, as I said before, for entry of the virus. So it's like a double whammy.
Yeah, well, we're gonna go back to some of this very shortly, but let me get to Doug Bingham. Mr. Bingham, it's really almost an undaunting task to be affiliated and associated with a world-renowned, preeminent research venue such as Scripps. Why don't we talk a bit about the width and breadth of the Scripps mantra and what they do relative to the research community?
Yes, it's Scripps Research is a very special place. We're an independent, nonprofit biomedical research institute, and our mission is basically to improve human health, train the next generation of young scientists, and educate students getting their Ph.D. So mostly it's a curiosity-driven endeavor. So we have across the institute we have approximately 200 faculty. About 50 of them are here in Florida, and we research all sorts of areas: cancer, infectious disease, neuroscience, which is becoming more and more dear to my heart in terms of why I can't find words, when I'm talking every day. You lose those nouns as you get older, and why do we do that? Okay, so neurodegenerative diseases like Alzheimer's and Parkinson's, basic processes, like why do cells that normally stay in one place in your body suddenly decide that they don't have to do that anymore, and they get out into your circulation and travel around and become metastatic cancers? I think Dr. Izard is working in that field. She heads up our cell adhesion laboratory here in Scripps, Florida. A lot of which is focused on, why do cells become cancers, and how do they metastasize? So it's a really broad range of endeavors of curiosity-driven research. It's a very interesting place to live and to work. Really, a lot of interesting people, as you well know. And when you're at an academic institution that does research, it's just a fascinating way to live your life.
Well, we thank you and to the leadership members of the Scripps Foundation for your presence here in the state of Florida, and really in the state of the world, because so much comes out of Scripps. Can I talk about something that just came to my mind? Do you want to talk about reframing?
Yeah, sure. One of the things we do at Scripps is we try to produce new technologies that help answer questions. There's a lot of questions out there in terms of human health, and sometimes they can't be addressed in very productive ways until you invent a new technology to do that. So one of the things that induced us to come to Florida and set up a campus here was the ability to build what was called an ultra high throughput drug screening, small molecule screening facility. And so we have a collection of about 750,000 small molecules that are potential new drugs. And there's a subset of those, it's about 12,000, that have already been through phase 1 clinical trials and proven to be safe in humans, but not necessarily effective for the diseases for which they were designed to use. Okay, so we've put together that 12,000, and we call it the Reframe library. It's a way to screen molecules that were already safe in humans for potential new uses. So we've used this, we've screened this library now for compounds that we know are safe in humans that may be effective in fighting the coronavirus infection. Someone who's already got the infection. Either in this, as we've looked at different combinations of those drugs as well as individual drugs. We've found about a handful of very interesting possibilities we are exploring with various pharmaceutical partners right now. So it's a very, very useful tool that we would never have had access to. The academic world would not have had access to had it not been for the state of Florida and Palm Beach County, providing us the resources to come here and build that.
This whole reframing thing is fascinating, but I understand that the Gates Foundation has been supporting the view of let's go back and we have this information, and maybe there's value to this beyond the pharmaceutical corporations or otherwise that were willing to take it on as a product. Not to demean or denigrate their efforts, but the Gates Foundation, I think, has enabled you to shall we say, enhance your reframing project. Is that correct?
Yeah. So what it allows us to do is, so when you go through clinical trials, the first thing you do is you got to test the safety of the new therapeutic drug, the new medicine in humans. Okay, and that's usually a fairly small trial. It's usually 10 to 20 people. You put it in, and you see, are there side effects? And if they don't have any side effects, then you have to put the new medicine into a much larger group of people to see if it actually has a therapeutic effect. Does it actually cure the condition or change the condition you're trying to work on? And sometimes what you find is, even though it's safe in humans, it doesn't have any side effects, it's not very effective. Okay. And so then the drug companies say, okay, fine, we're not going to work on that one anymore. We put it aside. But that process of getting it through phase 1 and then into phase 2 costs many, many, many tens of millions of dollars. So why not take advantage of the fact that there's a drug on the shelf that's been shown to be someone's already invested all those millions of dollars and it's shown to be safe. Maybe it might be useful in another disease. For instance, we're doing that now with a drug that its generic version is called ozanimid. It's known as Reposa. It treats multiple sclerosis. We had a hunch that maybe it might treat another disease too. And so we can go straight into a clinical trial without having to do phase 1 again and see if it affects Crohn's disease and inflammatory bowel syndrome and maybe some other diseases as well. So it's sort of repurposing the drug that was originally designed for multiple sclerosis. And so you save a lot of time and a lot of money and a lot of resources when you repurpose a drug known to be safe. And that's, so the Gates Foundation was very, very helpful in terms of providing resources for us to build out this collection. We call it the Reframe collection of drugs that have been shown to be safe, but not necessarily effective in humans. And we're just repurposing them. So it's a brilliant idea.
Dr. Izard, talk to me about this very unique microscope that you have, and also some of the outstanding work that's coming out of your area of structural and computational biology.
Yeah, so this microscope was a vision of many people, and one of them is Doug's as well, that it really changed, and my colleagues, for example, Dr. Farzan, which is a virologist here, that this microscope would really change our science, and it really already has in a very short time. It took me 2 years to install it, but because we can see now at an atomic level how things work, like a drug works, or understand or design a drug at an atomic level, like again, I said, like as small as water in 3 dimensions, that really has opened up so many possibilities for us in terms of attracting graduate students, for example, from different disciplines, and as well as, for example, in the drug design area and in terms of grant funding. Because, for example, I was able to get 2 National Science Foundation grant funding very easily with this new microscope being on board now, where before we are very heavily funded by National Institutes of Health, and National Science Foundation doesn't invest much in institutes like ours because we're so heavily NIH funded. And with this new microscope, we suddenly have been very successful suddenly to double the amount of grants that Scripps has with the National Science Foundation. So that's just is an endorsement how understand what impact this microscope will have on so many people, students, graduate students, faculty, interdisciplinary, back to making drugs and back to understanding basic biology at an atomic level. It's just really exciting time to be here right now.
Well, the amazing thing about the microscope, I'll give you a little brief history of vaccines, for instance. I think many of us remember the early days of the polio vaccine, and you had the Sabin vaccine, you had the Salk vaccine. Those are called those types of vaccines were whole pathogen vaccines. They inject the entire virus into your body. Okay, the Sabin vaccine was what's called a live attenuated virus, and the Salk vaccine was a killed virus. And there was a lot of controversy about both of those vaccines in the early days, whether or not people actually got polio when they got vaccinated. Was the virus, the Salk virus vaccine, was it actually thoroughly killed? And it turns out it was not at first. So, for instance, the Cutter Labs had modified the protocol for making the Salk vaccine, and it had failed to thoroughly kill the virus. So some people were actually getting polio from the vaccine. The same thing was true from the Sabin vaccine. It was an attenuated virus, but some people still didn't get full-blown polio, but they still suffered types of paralysis, or they were born with there were newborn babies with shrunken limbs because it wasn't completely attenuated. And so those are side effects of the vaccine of that type of virus. So what we've learned over the years as scientists is that the smaller piece, the less you use, the better when it comes to a vaccine. So you want to use, so for instance, if you were to, the SARS virus, the COVID-19 virus, has approximately 4 proteins on its coat. Your body makes an immune reaction against all of them, but it's really only making an immune neutralizing or protecting reaction against one of them. The virus also, makes probably another total of 28 proteins when it infects you. We don't need all those 28 proteins. We have found out that we only need the spike protein. And what you're seeing now is the vaccines that are the first generation of vaccines against the virus are all based on the whole spike protein. Okay. What we were able to figure out at Scripps, and in conjunction with using the microscope and other molecular biology techniques, is you really only, to make an effective vaccine, you really only need a tiny piece of the spike protein. And so when you use only the minimum amount required to produce a protecting immune response, you'll have far fewer potential side effects. And the fewer the side effects, the more accepting the public will be in terms of taking it. And that's really important when you're in public health and trying to get a large percentage of the population to take the vaccine. That these kinds of studies couldn't be done without the kind of instrumentation, the kind of scientists that Dr. Izard is, because they can look at the problem in terms of individual molecules as opposed to the large protein. So it's just so we can make now a vaccine with very few side effects, if any, in a matter of months as opposed to a matter of decades. That's all been the result of science of the past 60 to 70 years. Just truly been an amazing thing to see.
Dr. Izard, let me ask you, relative to what Mr. Bingham just said, what do you have to say about what he just said?
The microscope allows us to look at these interactions at atomic level. So it's a huge plus. We can really now move forward really, really fast. It's very, very good opportunity for us, an incredible opportunity for us here at Scripps, Florida.
The thing that we need to bring assurance, and the reason why I was anxious to have Scripps on is because we've had a lot of really requests over the years, but I didn't know how to fit it in. But this COVID crisis really was a perfect opportunity for me to bring Scripps forward and make people aware of the wonderment of the work that you all are doing. But let's get back to this issue of the inoculation and the products that are going to be created and have been created.
Well, public health is all about public education. If you're going to you can't have effective public health unless you have effective public education. And so I think one of the jobs we have as institutions of higher learning is to help educate the public that, there may have been problems with whole pathogen vaccines in the past, in the 70, 80 years ago. But we're at the molecular age now, where we have been able to pare down and design vaccines that have the absolute minimum required amount of foreign material used in them, so that you really don't get the side effects you had in the past. it's an education problem. Okay, it really is. But you're not going to get this virus under control until you have approximately 70% of the population immunized, one way or another. They're either going to, it will start abating when you hit that level. That's what's going to happen.
Dr. Izard, let me get back to you because we're down to the last 4 minutes of the show, and we really haven't brought the honor and praise to you for the work that's being done. And like I said, the issues that you are working with are so dominant to the care of multiple diseases. How proud of you of working where you are and receiving the type of support that you're getting.
I am extremely lucky to work in one of the top 10 graduate schools in the nation, and it's just extremely lucky to work on a very important cause and be part of helping so many millions of Americans and people worldwide. It's extremely a huge privilege. It's also a responsibility, but it's a huge privilege, and it's just an incredible opportunity. And my colleagues are just a very, very rich environment. We have Nobel laureates, we have National Academy members on our staff. It's just incredible. It's just, I can't even describe it. It's so amazing.
And it's people like you that we have to thank.
Thank you for the opportunity.
Well, back to you, Doug Bingham, because we're down to the last 3 minutes of this show.
I would just say that at the end of the day, science is a marketplace of ideas. And you have to listen to everyone's ideas, and they're all vetted, and they're all discussed, pros and cons argued. And at the end of the day, the strongest ideas, become what we accept as knowledge. And it's very collaborative. It's very iterative. It's a fascinating process to watch and to see and to participate in. And anyone who's thinking of pursuing a scientific career, please come talk to us. It's a wonderful life.
Well, we thank you both for representing, like I said, one of the preeminent research entities in the world, not only in the United States, but in the world. And like I said, I've been trying to figure a way to bring you all forward just to talk about Scripps. But this this insidious COVID crisis really, I guess you could say, gave me the opportunity to bring you forward, because I want people to understand that there are well-meaning people, both executive individuals, corporate individuals, as well as the supreme scientific minds and knowledge in the Ph.D.s and the medical professionals that are involved in creating new products for the behalf of our viewers and the multiplicity of people that receive the good effects and the goodwill of entities such as Scripps. So on behalf of the people that are viewing, I want to thank you. Doug Bingham, thank you very much for being here and being the boss of Scripps Research Florida. And Dr. Izard, I certainly appreciate your extraordinary accomplishments and knowledge. Thank you once again for being part of this broadcast, and I wish you nothing but good health and a lot of victories. How about that? Folks, this program, again, comes to you from Nova Southeastern University. If you have any questions or comments, there's an email address and a telephone number. And remember, I always tell you to take good care of yourself, but I want you to listen really very, very carefully, particularly what Mr. Bingham said, and what Dr. Izard also said. Vaccines today are quite different. Thank you once again for showing up. Thank you again, Doug Bingham. Thank you again, Dr. Izard. And remember, as I always tell you, take good care of yourself. But in this case, listen to what you heard, and when the vaccines become available, don't hesitate. Do what you think is right to protect you and your family. Thank you very much for showing up one more time and allowing us into your home. Remember, this program comes to you from Nova Southeastern University. My name is Fred Lippman. Until next time, see you.